Hi biologists, this is Miss R and we're going to do lab 3.17, feedback in biological systems. The first thing you want to do is pause the video and look around for the link to this MIT Open on feedback in biological systems. It's a very interesting 10-minute video and you'll need to know what's in that video to answer the rest of the questions in the lab. So pause the video and pause this video, go watch the MIT Open video, and then we can continue. Now that you know all about positive and negative feedback loops, we can talk about the differences. One of the differences is that negative feedback loops always are trying to get back to some sort of set point. They use different aspects of the system to always return to this set point here. Whereas a positive feedback system doesn't always come back to this set point, it kind of tends to escalate or amplify the response. Temperature, the way we have homeostasis for temperature in humans is a negative feedback loop. In other words, we're always trying to come back to 98.6. We're always trying to come back to that set point. What are some of the ways that your body maintains a constant internal temperature? How does your body always try to get back to that set point of 98.6 Fahrenheit or 37 degrees Celsius? You can think about it. There are a couple hints in this diagram. What do we do when it's cold out? As Minnesotans, you know the answer to this one. What do you do in the summer when it's really hot? It means you have to drink a lot of water. Those are some good hints. In the video, it talked about how childbirth is a positive feedback loop. And this diagram here obviously is not a childbirth. It's a different positive feedback loop, but it's a good way to illustrate how amplification works. Ethylene gas is produced by, say, one apple on a tree. That signals another apple to produce ethylene gas. Now there's more ethylene gas being produced, which signals more apples to be ripening, and those ripening apples produce even more ethylene gas. So the response keeps going up and up and up, and that's a positive feedback system. In the video, they talked about how childbirth is a positive feedback system. A woman's body produces oxytocin, it makes the uterus contract, and the uterus contracting makes more hormone. And so the response amplifies, of course, until the baby is born. Another example of a negative feedback loop is what you're going to find. We want one besides temperature regulation, which we talked about a few questions ago. Here's a big hint. Your blood sugar is something your body always wants to maintain at a fairly constant level. And we know from the video that diabetics, there's a mistake in that system because insulin is no longer recognized by some of the cells as a signal to pull sugar into the cells and out of the blood. Glucagon is the other side of the system. When you're feeling a little slow and laggy and your blood sugar is maybe a little low because you haven't eaten lunch in a while, your body releases glucagon, which tells your cells to release sugar out into the bloodstream, or your liver more specifically. Now that you know all about positive and negative feedback loops, it's time to think a little bit about population dynamics. There's a second video you need to watch. This one's only a minute and a half, and I'll put that link where you can find it. Now that you've watched the video on carrying capacity, hopefully you understand that this fishbowl is a little bit beyond carrying capacity. No more fish could live in this fishbowl, and that's really what a carrying capacity for an environment is. This fishbowl, on the other hand, you might be able to get a couple more fish in there before it reaches carrying capacity. Here's a good model for carrying capacity. These are things that cause populations to die off: starvation, predators, diseases, human development, loss of habitat. That's for perhaps a deer population here. And of course, annual reproduction is deer being born. And so you can see kind of these are the outs and this is the in. And so the population tends to stay at a kind of a level. It'll vary a little bit, but as the can demonstrates, there's a level which the population usually varies around. So this graph is from the video, and you can see that the population is kind of vacillating back and forth across a constant here. Does this represent a positive feedback loop or a negative feedback loop? And I'll let you think about that for a moment. Now that you know all about feedback loops, positive and negative, and you know about carrying capacity, we're going to talk about how these two factors play out in a lion population. I'm going to give you this URL and you can follow along, or you can just watch what I'm doing on the screen. So if you click on the URL I gave you and click the orange button that says Launch Activity, you'll end up on this screen here. And we're talking about African lions and modeling populations, and we're going to incorporate what we know about carrying capacity and positive and negative feedback cycles in biological systems. So the Ngorongoro crater in Tanzania, Africa, is home to many species of animals, including lions, zebras, wildebeests, and rhinoceros. As scientists, we can describe these groups of animals as populations. We've talked about populations earlier when we talked about evolution. A population is a group of organisms that belongs to the same species and lives in the same general area. Ecology is the study of interactions of populations with their environment. So which one of the following is an example of ecology? Studying earthquakes, looking for planets in outer space, creating pharmaceutical drugs, or studying how global warming affects whales? Make sure you answered the last question before you move on. You can see we're on screen 2 down here. In 1962, unusually heavy rains during the dry season caused a massive buildup of stable flies in the Ngorongoro crater. These flies drained blood and caused skin sores, which infected the poor lions and made them sick. By the following year, the population of lions had crashed between 75 to 100 to just 12. So it went from between 75 and 100 to just 12. And this activity will explore 2 different population models to discover the fate of the lion population. To answer the questions below, we'll use real data from Dr. Craig Packer's research on lions in the Ngorongoro crater. Which of the following questions interests you the most? And there's no correct answer to this question. You can choose whichever question you would like, and then click Next in the lower right-hand corner to continue. Populations change over time. Populations increase when more organisms enter a population, that's immigration, or when organisms leave it, emigration, or when more organisms are born than die. The opposite is true. Populations decrease when more organisms leave than enter, or more organisms die than are born. The data in this graph is hypothetical from 1950 to 1962. Researchers knew that there were about 75 to 100 lions in the crater, but they did not start collecting data until the population crashed here in about 1963. From 1960 to 1963, the population of lions in the crater decreased. Which of the following is most likely to have been true from 1960 to 1963? Well, let's look at the graph. Here's 1960, 1961, 62, 63. So we're looking at the orange part. Do you think more lions are immigrating, coming in, than emigrating, that's going out? Are more lions died every year than were born? No lions have died, or both A and B. Think about it. The graph is going down. What's happening in the population? Now we're going to look at a different part of the graph. Populations change, and we're going to look at this part of the graph here. This is the same hypothetical data from the previous page, the lion population from 1950 to 1963. However, since the lions are isolated in the Ngorongoro crater, let's assume that birth and death rates have a greater influence on the population than emigration or immigration. In other words, the crater is kind of closed off by high cliff walls, so it's hard for other lions to get in or out from other populations. So we're going to look more at birth and death rates. So what is happening here when the population is increasing? Are more lions being born? Are more lions dying? Think about it and choose your answer. What happened to the lions after 1963? After the crater lion population crashed to 12 individuals in 1963, what do you think happened? What do you predict will happen to the lion population after 1963? Explain below. Draw your prediction on the graph to the right starting in 1964. You may either click from point to point. Remember, your prediction starts in 1964. We'll later compare it to real data. So I'm going to let you choose an answer for what you think is going to happen, but I'll give you a hint what's going on in the graph here. Probably, since the stable flies are gone, we're going to see an increase in the population, unless maybe there's a drought or different disease. And I'm guessing the population might kind of return to what it was before. Now we're going to go from lions to bacteria for a moment to think about how populations grow over time. We're going to use a growth model to figure out what happened to the lions after 1963. However, we need to learn about growth models first. We'll use the growth model of bacteria as a simple model. Bacteria reproduce constantly. Let's say we put a single bacterium in a petri dish with some agar food. That's the gel-like material here in this dish here. These bacteria reproduce by cell division once per hour. Each time a cell divides, it gives rise to 2 cells. So what would happen over 12 hours? Well, if we started with 1 right here, after an hour we'd have 2, which barely registers when the graph goes up to 1,000. And then we'd have 4, and then we'd have 8, and then we'd have 16, then we have 32, 64, 128, about 300, so we're kind of starting to register on that scale, about 600, about 1,200, about 2,400, and then about 4,800. So you can see, if you remember from math, this is an exponential curve. It starts to grow very quickly. Wow, we did a pretty good job. Here is the actual graph compared to our prediction. Our prediction was in red and the actual graph is in blue. As you can see, the population starts off growing very slowly right in here, but then it starts to grow faster and faster right in here. Between 4 and 6 hours, the population went from 16 to 32. Well, between 11 and 12 hours, it went from 2,048 to 4,096. This type of growth is called exponential growth. In other words, the function will have an exponent, or one of those little numbers in the upper right-hand corner next to the variable. This is called exponential or a J-curve. This type of growth occurs when there's enough food, space, and other resources for every single bacterium. If we let this population of bacteria grow in the petri dish for 24 hours, the graph is going to look like this. So it starts out slow, and then it gets a little bit more, and then it just goes crazy. This is why you can get so sick from bacteria so quickly if they get really happy in your body. In which hour are the bacteria growing the fastest? If you're following along and doing this on your own, You actually have to click on the point to pick that point. So we talked about exponential growth in bacteria and how it increases very quickly, but lions are not bacteria. Unlike bacteria, lions cannot divide once an hour, but their population can undergo exponential growth. Ngorongoro crater is a 2,000-foot deep caldera with a 100-square-mile floor located at the eastern edge of Tanzania's Serengeti Plain. The crater's cliff walls serve to isolate about 100 lions from their nearby Serengeti counterparts, so there isn't a lot of immigration or emigration. Here is the lion population in the Ngorongoro crater. Hypothetical data from 1950 to 1962 is in blue. Real data from 1963 to 1980 is in orange, and our prediction is in red here. Looking at the lion population from 1963 to 1975, so the orange population, do you think this rate of growth will continue to increase? Why or why not? So think about this. It's growing really rapidly. It saw a rapid decline. It's growing really rapidly. Will it continue to climb like this for another decade or two? Limiting factors. The previous graph showed exponential growth in the crater lions from 1963 to 1980. However, exponential growth is rarely sustained for a long time in any population. A more realistic population model recognizes the factors that limit population growth. For example, the bacteria in our petri dish would eventually run out of food, preventing them from reproducing uncontrollably. Therefore, food is a limiting factor. Using the picture to the right to get started, brainstorm a list of at least 3 limiting factors that might affect the lion population in the crater. Well, the first thing I see here that we already talked about is food. Zebras are food for lions. So that's number 1. Food. The second thing you can think about is, where do lions, zebras, elephants, and other animals that live in the Serengeti Plain where it's pretty dry, where do they have to all go at least once every couple of days? Where do you have to go to tank up in the summer if you've sweat a lot? You get kind of thirsty. Think about it. What's a limiting factor for animals in a desert or in a very dry environment? So now that you've thought of your 3 limiting factors, let's talk about logistic growth. It's not exponential growth, it has a limit. The available resources in the crater can support a certain number of lions, just like the petri dish can support a certain number of bacteria. When limiting factors affect population growth, we describe it as logistic growth, or an S-curve. You can see how this curve here is exponential for a while, but then it levels off and it's kind of in the shape of an S. Both density-dependent and density-independent limiting factors influence the growth of populations. Density-dependent factors include food. There's less food if there are more animals. There's less food per animal. There's less space per animal if there are more animals. That's why it's density-dependent. Water, there's only so much water per animal, and the more animals, the less water per animal. Disease happens if there's lots of crowding. And predation, the more animals that are kind of living on top of each other, the more they're going to eat each other. For example, a large population of lions would probably decrease if the zebra died out. Well, a small population of lions would probably be okay. Density-independent limiting factors include weather. Tornadoes don't care how many animals are in a particular population. And natural disasters like floods or earthquakes. Click a point on the graph where you think competition for space was low for the bacteria. So this again is a logistic growth curve for bacteria, Where do you think they're not competing so much for space when there are not so many bacteria and there's kind of a lot of space in the petri dish? I'll give you a hint. It's pretty early in the history of the bacteria. I'll let you take a guess. Food is a good example of density-dependent limiting factor that could inhibit population growth. What do you think will happen to the rate of lion population after 1980? Will it grow faster and faster or start to slow down? Remember that the lion population was between 75 to 100 individuals before the disease outbreak. When we know about where the population hovers, what do we call that when we watch that second video? Take a minute to think. Use the graph to draw your prediction about the lion population from 1980 to 2012. So I think that 75 to 100 is going to be the carrying capacity. So that the lion population is going to fluctuate, but it's always going to come kind of between 75 and 100. So it might do something like this. Let's check. The number of individuals an environment can support is called the carrying capacity. So that 75 to 100 was definitely the carrying capacity. The limiting factors determine the carrying capacity, which is generally the highest population that the resources, food, space, water, can support. Competition for resources is highest when the population is near capacity. Populations usually grow exponentially in the first half of the logistic growth and then stabilize as carrying capacity in the second half. So here's where the exponential growth occurs, and then it slows down and you kind of reach carrying capacity here. This is the petri dish, has no more space for any more bacteria to grow on the agar. So here we're going to draw a line to estimate the carrying capacity of the petri dish. To draw a line, we're going to click 2 points on the graph. Afterwards, drag and drop your 2 points to edit your line. The coordinates for the 2 points in the table will be shown below. So the coordinates, the X and Y coordinates, will be shown here. I think it's right around here, so I'm going to draw a line from here over to here. And we'll check my answer. Lo and behold, I was correct. The carrying capacity of the petri dish is about 6,000 bacteria, limited by food and space. Here are two data sets from 1952 to 1950 to 1962, that's in blue, and from 1963 to 1980. Remember that the first set is a hypothetical carrying capacity, while the second one, orange, was collected by scientists in the field. On the x-axis, year 0 represents 1963, when the study began. Draw a line to estimate the average carrying capacity of the lion's environment prior to the disease outbreak that took place in the 1960s. So if we're going to look at the carrying capacity in the environment prior to the 1960s, so right in here, I think the average is going to lie right in here. So I'm going to click from here to here. Let's see if I'm right. Yeah, wow, I was right on. Cool. So this is what they consider to be the carrying capacity of the lions before 1963, before that disease outbreak. Here's our prediction about the lion population from page 11. Does our old prediction does it look different than what we might have been expecting? Use what you know now about carrying capacity, redraw a new prediction of the lion population after 1963. Well, I still think it's going to be around the carrying capacity, but maybe it'll be a little bit lower because the carrying capacity was between 75 and 100. So I'll draw my curve maybe a little bit lower. And I know climate change is affecting Africa, so that might be a factor as well. Here's the actual data from the next 20 years. As you may notice, the real population data isn't as perfectly shaped as the bacterial population growth we saw. Real populations often fluctuate as the limiting factors change from year to year and may stabilize after several generations. Wow, it kind of sank a little bit more. I wonder what caused the decline here. So compare my prediction to the real data. What do I notice? Are there differences? Can I suggest some causes? Well, it's a little bit lower. Maybe drought had affected the crater, or maybe there was another disease outbreak. Or maybe there were some lion poachers that came in and took a bunch of lions. I don't know. Let's find out. The population decreased below the 70 to 90 lion average carrying capacity of the crater before 1963. As previously mentioned, density-dependent limiting factors were thought to include food, space, shelter, water, disease, and predation. The limiting factors that determine the carrying capacity of the crater may be different than they were 50 years ago, thereby changing the carrying capacity. Which of the following is a density-independent limiting factor that could cause fluctuations in the carrying capacity between 1995 and today? Well, I mentioned it earlier. Whether it rains or not doesn't matter how many animals are in a population doesn't affect whether it rains or not. So that is a big hint in choosing the correct answer there. Now let's apply what we've learned about bacteria and lions population growth to humans. Here are 3 different growth curves for human population growth. This is the highest model predicted by the United Nations. This is kind of the medium growth model, and this would be the lowest United Nations predicted model. Given the shape of these graphs, do you think Earth's population will reach a carrying capacity? What might be some limiting factors? What do you think will happen if we do reach carrying capacity? So this kind of requires you to pause the video and do some thinking. It depends on which model might be correct. For this model, I think we definitely would reach carrying capacity if we continue to grow at that rate. That's a little bit scary. We might run out of some things like food or water or space. Congratulations! You've learned about exponential growth, logistic growth, carrying capacity, and density-dependent and density-independent limiting factors. You made predictions, interpreted graphs, and applied population models to real data sets. You're now prepared to answer the questions from the beginning of the activity. Which one did you choose? What factors can influence how populations change over time? What is the difference between exponential and logistic growth? How can we apply population models to real-world data? That was the UN data we just talked about. What inferences can we make about the human population? So take some time, do some thinking, and write your answer. Now that you've done some thinking about population models, we're done, except there's one more thing you need to think about. Fortunately, we live in Minnesota, and we have Dr. Craig Packer from the University of Minnesota who did this research on lions right here. I believe he works on the St. Paul campus of the University of Minnesota, which happens to be pretty close to my house. If you're interested in learning more about Dr. Packer's research, you can check out the article from National Geographic. There's also a scientific paper you can look at. Thanks so much for watching.